We are starting. Okay, so again, welcome everyone to my talk on experimental design. I called it Experimental Design Matters, which I kind of ripped off that title from the chapter by the same name from this stats book that I really like. It's kind of got a double meaning. Like, we're going to talk about matters of experimental design, but also it matters how you design your experiments, because how you design your studies influences the quality of the answers you get to the questions you care about, right? So that's why we're here. Here are a couple of take-home messages that it would be good to get from this talk if you don't pay attention to the rest of it. These are kind of the main points I'm trying to hit. I'm not trying to get too in-depth in the technical aspects of this, since this is only a short talk. This is more big picture concepts and that kind of thing. So yeah, the main point I'm trying to make here is that an experiment is a model, like not necessarily in the technical statistical model sense, but it's a model, like it's an abstract representation, an imperfect representation of the real world. It's not real, just like our statistical models aren't real. Experiments are kind of not real either. They represent the real world that we want to learn about. But through this controlled little microcosm, we can understand stuff about the real world and cause and effect that operates there from that. And another major point I'm going to hit over and over is there's no one size fits all best experimental design. It depends on the question you're trying to answer, and there's trade-offs, right? You can improve one aspect of it, another one will get worse as a result. That's always something to keep in mind. And lastly, I already said this, but proper experimental design is really crucial because it can help you maximize the ratio of signal, so variation due to the thing that you are interested in, to noise, any other variation you don't care about, and that will help you do better science. So keep all those things in mind as we have our little conversation. And again, please interrupt, either just unmute or put it in the chat if you have a question. So, here, we'll start with a little Q&A for you all. I'm just curious, can anyone give me, just off the top of their head, a definition of experiment for me?
It's a way to test a hypothesis.
Excellent. That's a very important part of it. Does anyone want to add to that, or are we all happy with that? Okay, we'll go with a way to test a hypothesis. So yes, testing or comparing hypotheses is one crucial part of it. Another is kind of this element of a controlled procedure. It's not just it's different from making observations. Could also be used to test a hypothesis, but we're actually controlling or manipulating conditions to test a hypothesis. And yeah, so it's a controlled procedure. We use it to gather evidence to either support or refute evidence against a hypothesis, or to compare between multiple hypotheses, or you could sort of use it to demonstrate a already known fact as well. And the idea behind experiments is that they give us information about causality. When we change one thing, what happens to another thing? And experiments are part of, the scientific method is more complicated than this, but if you sort of oversimplify things, you could say, well, we start by making observations of nature. We use those observations plus other prior knowledge we may have to generate a hypothesis. And then we use an experiment to test that hypothesis, and then the cycle repeats itself. I mean, obviously, this is a little oversimplified. There are some scientific disciplines that cannot do experiments. Like, what if you're a cosmologist studying the early universe, or like a paleontologist or something? You're not doing experiments to see what would happen if evolution happened a different way than it did. You can't really do that. But in the fields that we work in, there's plenty that we can learn from experiments, and they're a cornerstone of the scientific method, at least in our little part of it. Now, I just said before, an experiment is a conceptual model, an imperfect representation of reality. It falls along a continuum of models of reality. Now, the famous statistician George Box said, as part of a greater quote, but he said, all models are wrong, but some are useful, right? So every model, be it an experiment, a simulation, a statistical model, is an imperfect representation of reality. It's going to leave something out. But some of models are useful, meaning they, through their abstracted simplification, they tell us something about reality, right? So it's no different than experiment is just another way of doing that. Now, experiments, I've made sort of a axis between greater realism and greater control, right? So like observing things in their natural setting is maximal realism. Those data points are the closest to real world conditions, right? Versus, let's say, the highest controlled conditions would be a simulation, with a set of mathematical equations that we use as a model, right? So that's not as realistic, but it's sort of the most controlled. Experiments are pretty far along that control side of things. We sacrifice realism. We're getting away from real world conditions to be able to have greater control of the variables in the system. It's a trade-off. So it's just somewhere along that trade-off. Now, there are so-called natural experiments, which is like taking advantage of pseudo-random things that kind of resemble an experiment that happen in the real world that could kind of get you halfway between a truly randomized experiment and observing the real world, which are also very valuable. And they fall in between pure experiments and observations. But again, there's sort of issues there. You have to really be careful making sure that it truly is close to random how the different conditions shake out. And we'll talk about randomization as we go in this talk. So before we get into the experimental design, this isn't really that important, but I just think it's cool. I just have a few slides about the history of scientific experiments as we know them. Now, obviously, every human being that's ever lived has done experiments, right? Young babies, as young as a few months old, will consciously manipulate things in their environment to see what happens, right? And that's how they learn about the world. That's experiment, just the same way that we do it. So this isn't the history of all experiments, it's the history of sort of formal scientific experiments as we know them, because we humans are just born to experiment. So the first sort of scientific experiment as we know it, usually people trace that back to about 1,000 years ago in the Middle East. A very intelligent person named Ibn al-Haytham did experiments with lenses and mirrors. Now, at the time, this may seem silly to us now, but it was thought that the way vision worked is that light rays went out of your eyes and hit the things that you were looking at, because that's what Aristotle and the ancient Greeks said. Well, this guy decided to put that to the test empirically by manipulating lenses and mirrors and test that hypothesis, and did a lot to advance our understanding of optics, especially the sort of theory that it is actually because of light rays going and hitting the lens in your eye that's how we see. And experiments progressed over the next few hundred years. Another notable milestone was in the 1600s in Italy. Galileo did a series of experiments on, he built contraptions like the ramp in this, it looked similar to the ramp in this picture, and he rolled steel balls down the ramp, timing them with a pendulum that he calibrated using his own heartbeat, and testing hypotheses about how forces like gravity act on physical objects. And another really important type of experiment is randomized clinical trials in medical science. And I think this is kind of cool. Now, this story may not, a lot of times these history of science things, they oversimplify the story and make it seem a lot cleaner than it maybe really was in reality. So maybe this isn't really how it went, but this is how the story has been passed down, is that, in the 1700s, scurvy was a huge problem in in sailors in the British Navy, and they didn't know any effective treatment for it. There was a lot of kind of folk remedies out there. So a ship's doctor named James Lind in 1747 did what we now consider to be the first randomized controlled clinical trial, where he took 12 of the sailors on the ship and split them up into 6 groups. Each group had 2 people in it, and each of those groups got a different treatment. And some they gave cider, some had to drink sulfuric acid. I guess they thought that would help. Some drank seawater and so forth. Now, we now know the mechanism of why scurvy happens, which is it's a vitamin C deficiency. And so some of the lucky sailors got to eat some oranges and lemons every day, and it is said that they quickly recovered from their scurvy, and this was seen as evidence of, this experimental treatment, right? Because it had randomization, it had a little bit of replication, and it was a controlled, like the conditions were equal for everyone other than the treatment they got. So it kind of fulfilled the principles of a good experiment, which we'll talk about more in a bit. And, that was kind of a little bit of a one-off thing, but really it's in the 19th century, the late 1800s, that the formal principles of experimental design were set down in a systematic way, basically the way we still do them now. And another big landmark was kind of in the 1880s. Charles Peirce, a kind of Renaissance man / genius, did a randomized controlled trial in psychology to test how sensitive people's hands were to pressure, and used a randomized trial with a repeated measures design for that. So that was an early example. Obviously, another major player in the early days of experimental design was R.A. Fisher in the 1920s. He wrote some hugely influential papers on agricultural science, experimental design, that basically we still follow a lot of those principles today. That's why, right, we say plots and blocks, and we use that kind of lingo. Well, even non-agricultural science uses that terminology, even if they don't have actual plots of land like we do, because of this pioneering work that was done by R.A. Fisher. Well, the modern era of experiments, we can kind of, not counting the sort of outlier scurvy one, the first modern randomized controlled trial with the proper placebos and all of that kind of stuff as we do it now was done in 1948 on a drug trial of antibiotics to treat tuberculosis. Now, before that, right, medical journals pretty much only had anecdotal studies where, a doctor would write up a case reports from a few of his patients, probably very rarely her patients, and write up his opinion about it. And that's very useful. It's incredibly important. We still need those kind of studies today, but before randomized controlled trials, that was all there was, right? And that's it can be seen that there are some huge biases in doing all of medical science that way. So it's very important that those are only complementary to randomized controlled trials nowadays. And at least in the field of industry, a lot of the experimentation approach that is done now was pioneered in Japan in the post-war era. They set up a lot of techniques for experimentation for quality control of their products. So we sort of had these parallel developments in, agriculture, in psychology, in medicine, in industry, that have all kind of now more unified into an overall framework of how experiments are designed. And I just threw this point in as well, nowadays some say, experiments are not as important and as useful as they used to be, right? Now we have big data and we have AI and so forth. Why do we need to do these small scale, very resource intensive, manipulative experiments? We can learn everything we need to know without them. Well, that's a bit of a straw man argument, but I would say there's definitely that vibe. And I think it's important for people like us that do experiments to emphasize that they still have an important role. There are certain types of knowledge you can only get from doing manipulative experiments, or you can best get that way. And so we'll talk about that as we go on. Does anyone have any questions or comments before I go on? Okay. So that was kind of the fun history lesson. Now we're going to go and systematically go through the basic concepts and principles of experimental design in the next part of this talk. So here are some of the little lingo that I'll be throwing around. I'll define these terms as we go. So, okay, treatment, right? That's a term that that people have heard a lot. Well, what is a treatment? It's anything that we, as the experimenters, manipulate or impose on the subjects of the experiment. And factor is any variable where the levels, the different values it can take, are set by the experimenter. So different treatments could be considered different levels of one factor. Like, for example, different doses of a medication, right? Medication is the factor, and the different levels of dosage are the different treatments, or different qualitatively different management methods for an agricultural field might be different treatments of the management factor. And what is a control in the context of experimental design? A control is any observation within a study that shows the effect of no treatment, or of a sham or placebo treatment, a treatment that we think has no effect, and that is used to establish a baseline that we can compare the treated group to see what the effect of that treatment is relative to if it didn't, was not there. Now, what constitutes a control? Well, that depends on what you are controlling for, right? Like if you had a vaccination study you were doing, let's say, and you have to administer that vaccine. Let's say it's an injection, you stick a syringe in there. Well, you could control for the effect of actually sticking in the syringe, right, by sticking in a empty syringe or a sham syringe into some of the control individuals, and that would control for whatever effect of getting stuck with a needle has on you. Or you could have some control group that didn't get the needle or the drug at all, right? And that would be a different control group. So it depends what you're controlling for. And so ideally, all variables that you manipulate in an experiment would have a non-manipulated counterpart in the control group or groups, right? Like the vaccination example, we're manipulating whether you get the vaccine. We're also manipulating whether we stick a needle into you. It's kind of an unavoidable part of the way the experiment is done. So hopefully you would control for all of those aspects. And you may have heard the term positive and negative control. What does that mean? Well, a negative control would be a baseline to compare the treatment effect to, right? Like if the treatment is vaccination, it would be an unvaccinated group. Positive control would be controls that we think will have an effect that will ensure the procedure is giving expected results using standard methods, right? Like if we are testing an experimental vaccine, we don't know how well it works, we would have a negative control, unvaccinated. You might also have a positive control, a already proven to be effective vaccine that you could also compare your new method to see if it works as well or better than what we consider to be the current standard. And in some designs, there isn't a separate control group with different individuals. Sometimes there's sort of a pre-post design where we take pre-treatment measures on a set of individuals and then do the manipulation and look at their measure the outcome post-treatment and compare them within. So that's another way of controlling for unwanted variation. Obviously, especially in human subjects research, where if we really know that a treatment is going to help people, if we really think it's helpful, it's sort of unethical to have a control group that you give no treatment to, right? And so you would need to do something like this to a within subject pre-post design that would be more ethical. Okay, experimental unit. This is an important concept, and it's one that I think a lot of people kind of don't have clear concept of when they're designing experiments. So let's think about what is an experimental unit. That is the thing or the object that we independently, randomly assign the treatments in an experiment to. And right, like if you're doing a field trial where you're planting out different genotypes, the experimental unit is the plot of land that you randomly assign one of the genotypes to be planted on. That's the unit. Or if it's a vaccination trial, it would be whatever individual plant or animal you decide to give the vaccine or not to. And experimental units can be really big, right? They can be very small or very big. I put this picture of, little flasks, right? Each flask has a few milliliters of fluid with some E. coli cells in there, and we can administer treatments to these flasks, right? So the experimental unit is the flask and whatever cells are in it. But it can also be as big as an entire drainage basin, like our colleagues in the Forest Service at Coweta Hydrologic Lab have a very long-running experiment where they manipulate entire watersheds, right? It's like a basin about a kilometer square with some streams coming out the bottom, and in some they clear cut the forest, and some they leave it intact, and some they selectively log and then look at the, water chemistry and soil chemistry and all that kind of stuff, right? But it's that entire watershed with all the trees in it that is randomly assigned to be logged or not. So that's the experimental unit. Now, the experimental unit, so we have a question from Lena. I can't really see it in the chat. Can you just ask it?
Yes. So my question is, can you have, for us, we're plant or fungal biologists, so I'm going to put it in that context.
Right.
So can you have a plant be both controlled and treated and still consider that plant as a single experimental unit?
Well, so in that case, no, the experimental unit would be, right, like if you treated one leaf or whatever, or one branch and left another branch as control, it's the leaf or the branch as the experimental unit because it was randomly chosen to be treated or not. And so that is the unit at which the randomization takes place, and that's the experimental unit. The plant is actually a block, a set of experimental units that are connected or related to one another, which we'll talk about in a minute. But yes, so the experimental unit, if there's multiple treatments within that plant, the plant is not an experimental unit. It's multiple experimental units connected to each other, right? Because the randomization is what determines the experimental unit. And on my slide, I have that the experimental unit is not necessarily the same as the observational unit. The observational unit is what we're making measurements on, right? Like, let's say the watershed, number 10 had 3 streams in it, and we measure the chemistry of each of those 3 streams. Well, those 3 streams are not independent because they're all part of the same watershed that was given a treatment. And so we've taken those are observational units. We've taken multiple measurements of different observational units within the same experimental unit. And we would have to account for that in our statistical model. That's where blocks and all that stuff come in. And I'll talk about that as we go. Great question, though. So let's keep talking about how we can break down experimental design into different components. Well, it's usually thought of as having 3 components of any experimental design. One is treatment structure. That is, what are the treatments and how they relate to each other. I'll get back to that in a minute. Design structure is how the treatments are assigned to experimental units, how the units are structured in space and time. That's where you say, well, what are the spatial layout of the plots? Like, is there blocking and so forth? That all goes under design structure. And response structure is what responses will we measure and when, where, and how in relation to the experimental unit. So that would be like, well, if we're going to do repeated measures over time or multiple measurements within the same experimental unit, that's all part of the response structure. Those are all sort of semi-independent things you have to decide about when you're designing an experiment. Some of the things are kind of, you choose them because they're the best for your question, and some are determined by the constraints of the system you're working in. And all of this stuff applies as well to observational studies as well as experiments. So even though we don't randomize in an observational study, we still have to decide the spatial layout of the units we're going to be observing, and selecting a gradient of natural variation, and deciding when and where to measure everything. So all these questions still apply even if it's not a randomized experiment. So I mentioned
Quentin,
I'd get back to
I got a question about that observational, like your previous slide. So when you say observational studies, do you mean like the difference between like you're taking a picture of like something that is fluorescent versus I'm quantifying like growth or something like that?
Well, an observational study, I would mean that anything where we do not experimentally manipulate the variable of interest, right? Like if we if you like went out into a field and found some diseased plants and some not diseased plants, there were naturally some, we don't know why some of those plants were diseased and some were not, right? So we can measure stuff, we can measure variables on those plants, but we didn't actually manipulate, versus taking some plants and artificially inoculating some of the plants with the disease so that they were randomly given that disease and then measuring the outcome, right? So the difference is, in our field observation, there may be other confounding variables, like maybe, weaker plants that are less biomass or something already get the disease. And so there's confounding variables, right? So the difference between the experiment and observation would be, are we manipulating the variable, right? Like you could do observations of fluorescence of whatever in that experimental context or in the observational context. But what matters is whether you manipulated the variable of interest artificially or not. Okay, so treatment structure, that would be things like, you may have heard one-way design, like where you have one factor with different levels, a factorial design, a regression design where you have a continuous variable with different levels that you're manipulating. There's other more complicated ones. Very common type of design that I see a lot in ARS is factorial design. That's where you have multiple factors with different levels, and your experiment includes all combinations, or at least a subset of combinations of levels of those factors. Like any type of G by E interaction study is usually a factorial experiment, right? If you want to know the interaction between genotype and environment, you're going to need multiple genotypes and multiple environments, and include all combinations of genotype and environment in your study so that you can estimate whether different genotypes express different phenotypes in different environments, right? That's G by E interaction, very common type of factorial design. Now, obviously, you don't necessarily just have to do 2 factors. You could do 3, 4, even more. The more interacting factors you include in an experiment, some would argue, well, it's more realistic, right? Because you have, in reality, it's not just one variable that's changing, it's many variables changing at the same time, and we do need to know how they interact. So it is more realistic to set up a complicated experiment with many interacting factors. But of course, it's the curse of dimensionality, right? Like if you had 2 factors with 2 levels each, there'd be 4 combinations, right? But if you had 3 factors with 2 levels each, it's 8 and 16, and it blows up exponentially. So you need way more samples to do a high order interaction experiment. Plus, it becomes harder and harder to interpret what does like a 4-way interaction mean, and so, right. Again, it's trade-offs, right? We're trading off realism and kind of complexity and ease of interpretation there. Something to keep in mind when you're designing an experiment. Okay, so what are the basic principles of experimental design? These are the three that were outlined by R.A. Fisher in his pioneering work on it, and we still follow these today. Randomization. We must randomly decide which units get which treatments. It's not a controlled experiment if you don't, right? Because if you don't, you might overestimate or even underestimate the effect of treatments because there's going to be confounding variation. What we want is to hope that that it doesn't matter which unit in our experiment was assigned to treatment and which was assigned to control, that the average response should be the same. If we happen to switch around which plots got treatment and which got control, by chance, we should get the same result either way if our experiment is properly set up. Replication is also important. We need to estimate the variation within treatment versus between treatment. In order to know what the variation within treatment is, you need multiple experimental units with the same treatment. You might need a lot in some cases, right? And that's how we can't know whether the average response is the same or different unless we have replication. And finally, the third principle is local control. We want our conditions to be as consistent as possible across the treatment groups. Again, this fits where we want to get rid of any other source of variation as much as possible besides the one that we are experimentally manipulating and care about. And this can be done to a great extent if you have good experimental protocols and have everything clean, then that's going to minimize the influence of those extraneous variables. Of course, we have statistics that can help us account for any other variation that we can't completely eliminate, but it's better to have as good of protocols as possible to minimize that variation so we don't have to do super fancy statistics to pick out the signal from the noise. We want to reduce the noise in the first place, right? Yes, Lena.
Christine, going back to your previous slide, and I don't know if you're going to talk about this, but I'm curious. Would you be telling us a little bit more about randomization if you have like a block design versus
that's
like a
basically the rest of the talk. So let's yes, I'm hoping, I think we still have 20 minutes, so I should be able to get through quite a bit of it. But yeah, that's basically the rest of the talk. So yeah, what I'm going to talk about for the rest of the time is kind of a little bit more technical of experimental design, how you would do it in different contexts, and which design is the best for a particular situation, as much as I can in a few minutes. So what we want when we're designing an experiment is the optimal design. That is, we come up with some criterion from a statistical model, like estimating the difference between two means or estimating a slope or whatever. And we can come up with, given the amount of resources we have and the number of units we have, what is the best possible experimental design to estimate that value. It's called the optimal design, right? Given the resources we have, we want to maximize our statistical power, our chance of getting a reliable answer to the question, the specific statistical and biological question we care about for the purpose of this experiment. And you might see the word efficient in the literature. Efficient, in this sense, refers to having the highest power for a given number of experimental units. That is the most efficient experimental design. And it's the most efficient or optimal design is the one that gives you the highest ratio of signal to noise, variation we care about to variation we don't care about. And if we have that ratio as high as possible, we have the highest statistical power to get an answer, right? Yeah, so we want to, again, the criteria that we are trying to minimize is we want to minimize as much as possible both the bias, we want the most accurate estimate, and we want to minimize the variance. We want to reduce the noise around that estimate as well. Now, again, as I already said and will say again, not one design is optimal for every situation. It depends on the specific hypothesis you're testing. I'll get into that a bit as we go. So to give you a little bit more concrete examples of what the heck I'm talking about, I'm going to have a few slides here where I'm going to go through kind of common experimental designs and their pros and cons. So I'm going to conceptualize this as, let's say we're just doing a field trial where we have a big area of field and we're splitting it up somehow and assigning treatments to different plots in the field, right? That's how we'll just use that as a starting point. Well, a very commonly used design is the completely randomized design. And it's sort of the simplest one, right? We just completely randomize every plot independently. There's no grouping of experimental units. Like here, in this example, let's say we had 3 genotypes and 2 treatments, right? That's 6 combinations. And we're going to do 4 replicates of each of those 6 combinations. So we have 24 plots in our field. Well, the CRD, completely randomized design, would be just totally at random, assign each plot to a treatment combination and do each one 4 times, right? Now, this is decent, but the problem is it doesn't do great with local control because we have, right, in our model, we would have like a term for genotype, a term for treatment, a term for their interaction, and an error term, which would have all of the extra noise. Well, that error term includes variation due to treatments, but also variation due to the environment. We can't pull them apart because we don't, there's no way to tell apart treatment variation from environmental variation. So this design is good if you have very highly controlled and very homogeneous environments, like a lab or greenhouse environment, like with lab rats or whatever. Each rat is in its own cage that's exactly identical. And so there's not really like that much environmental variation to begin with. So there's not too much environmental variation to care about. So you wouldn't have to worry that much about blocking in that case. But this is not the most efficient design. It does not have the most statistical power for most field experiments where we actually do have oftentimes natural variation in the field. There might be a gradient of soil fertility or, light availability or whatever in this field, which would be confounded with the other variation if we don't use blocking. Oh, yeah, I forgot about this point. There is a way, even if you do have a CRD to control for the spatial variation, we sort of can use a continuous spatial effect in our statistical model to control for it even without blocking, which is great, and I often do it, but blocking is still a great way to deal with some of this variation. Yes, Lena, I'm going to talk more about blocking in a second, but do you have a question, Lena?
Yeah, and I don't know if you're going to talk about this, but like, for example, like if we have, you're talking about like light resources and stuff like that, like in a greenhouse.
Yep.
Depending on where you are, you're gonna get differences in light.
Right. So even right. Yeah. Even a greenhouse is not homogeneous, right? It's not as bad as being in a field, which has topography and all this other stuff. But even a greenhouse has internal environmental variation that we want to account for as much as possible. So hopefully as we go through the next few slides, you can see how we might do that. Right. So what about blocks, right? I keep talking about it, but let's actually sit down and define what is a block. That is any group of experimental units that are somehow related or connected to each other. That could mean they are closer together in space or closer together in time, right? If you know the first law of geography is all else being equal, the closer together two things are, the more similar those two things are, right? They share environmental influences. The more closely they are together, the more similar their environments. And we can take advantage of that as we set up experiments. We can use this idea to account for other influences in the environment besides the one or ones that we are experimentally manipulating by grouping close together units into blocks. And a block design for the same number of units can actually have higher statistical power to detect effective treatments than a completely randomized design. Like, for example, right, like in our field example, we could divide up our field into small units of plots that are compact and close together. and each of those groups of plots would be a block. Or if you're doing a study on educational interventions, right, you would do it at multiple schools. Each school has multiple classrooms. And like at school A, you would do one intervention at one classroom, another intervention at another classroom. And the classrooms are the experimental units that are assigned to treatments, but the school is a block, right? The classrooms within the same school are related to each other. They share the same influence, they have the same principal and, you know, all that kind of stuff. So the same part of town, so they're correlated. Or if you did a study of a drug and you did it at multiple hospitals, right? Some patients at hospital A got the drug, some got the placebo. Well, each hospital is a block, right? Because all the patients in that same hospital are sharing the same influences and therefore should have correlated responses to the treatment. So that would be a block. And we would account for that in our statistical model accordingly. So the most basic type of block design there is the randomized complete block design, where each block is a complete replication that contains every treatment combination exactly once. So like in our 3x2, 3 genotypes by 2 treatment studies with 4 replications, this is how you do it as an RCBD, right? Like each of these rectangles contains all 6 treatment combinations set out in a completely random way within that. But we set up the randomization so that each of these blocks gets exactly one of the treatment combinations. So every block has all 6 of them. That's an RCBD. Another type of randomized complete block design is a split plot design. And that can be done if you have 2 or more factors, then you would have 2 or more levels of randomization. So one of the treatments is randomized at the main plot level, the other at the subplot level. Now this is, so here again, we have a complete block, like this quadrant of it has all 6 treatment combinations, but we split our block in half into what we call main plots, and we assign, like here, this main plot got treatment 1, the circles, and this main plot got treatment 2, the triangles. We chose that randomly, and then within that main plot, we randomize genotype at the subplot level, so that we have each block has half of it is treatment 1 with all the genotypes, and the other half has treatment 2 with all the genotypes. This is still a complete block design, right? Because all treatments appear once in every block, but in some cases, like in field studies, it might be logistically difficult to randomize treatments at that fine of a level, right? If it's some management treatment that you can't do a different management treatment on tiny little plots, you might randomize the management treatment at a larger level, but you can sew genotypes into small little plots very easily. So you would randomize genotype at the subplot level. So this can be done for logistical reasons, but it also can actually be a better design than RCBD for some things. So like compared to a randomized complete block, this split plot actually has higher power to detect a subplot treatment effect. So that would be like to detect the genotype effect, it actually has higher power than the randomized complete block design. And the interaction effect, it has a higher power as well. But it has less power to detect the main plot treatment effect. Like in this case, right, we don't have as many independent randomizations of the T treatment factor. We only randomized it 4 times at the main plot level, right? There's only 8 main plots there. And so we have less power to detect that main effect, but more power to detect the subplot effect relative to an RCBD. So again, it's these trade-offs I keep talking about. Okay, so yeah, I don't want to go too over time, but I'll do a few more slides. You can like stop me if we're getting too close to the time. But here's what we statisticians always joke that ARS scientists love RCBDs and always are setting up their studies as randomized complete block design and never consider any other alternative, because I guess they learned in their, one semester of stats that they had that RCBD was a good design, and so they just go with that. Well, yeah, they're decent and they do fine in a lot of situations, but that is not all there is. And in fact, they may be objectively worse than other designs and have less power for the same number of experimental units. So, you shouldn't just blindly like set everything up as a randomized complete block design. You should actually think about or do an analysis to look at what the highest power design is for your situation. So I don't know if that blew anyone's mind or what, but I'll talk about a couple other designs here real quick. So, right, we've only shown complete block designs so far, where each block has every treatment combination. Well, what about incomplete block designs, right? That would be any block design where only a subset of treatments are in each block. Each block, right, if you have 6 combinations, like each block might only have 3 or 4. It doesn't have every possible combination in every block. There is no law that says blocks have to be complete, right? Because the idea of blocks is that they are environmentally homogeneous areas. Now, if you are forcing yourself to do complete blocks, who is to say that the size, that an area that's maximally large, that is environmentally homogeneous, that's more similar within it than to anything else, is exactly the same size as the area that fits exactly one plot of each treatment? There's no reason why those two things have to be the same, and they're often not. Like, especially if you did a breeding trial with hundreds of genotypes, right? Like, complete block would be huge. It would have one of every one of these hundreds of genotypes. That might be too big to actually be environmentally homogeneous. So block is no longer working for what it's meant to do, which is to control for that environmental variation besides the thing that we're manipulating. So why should you force yourself to do complete blocks there anymore? There's no good reason to do that. So, and again, like I said, you might have to make incomplete blocks, but you also might get more statistical power. They might be more efficient. You don't know until you look in your particular situation. So a very simple type of incomplete block design is the balanced incomplete block design. And that would be one where every treatment appears equally often in the same block with every other treatment. Like in this example I made, like here you can see treatment one time it's in a block with 4 and 7, one time it's in a block with 3 and 6, and one time it's in a block with 2 and 5. You can look at all the other treatments and see that every treatment appears once with every other treatment, and it's perfectly balanced. Now, this may not be possible depending on exactly how many experimental units and blocks and treatments you have, but you can use software algorithms that will output you a design that is as close to balanced given your parameters as you can get. And so these actually are reasonably efficient designs. And yeah, so in this particular example, each treatment is in a block once with every other treatment, and we have 3 replicates of every treatment, right? There's 3 1s, 3 2s, and so forth. So that's an example of a incomplete block design. Another type of incomplete block design you may see is the disconnected incomplete block design. You can see that each block gets a treatment set, right? Some of the blocks have treatments 1, 2, and 3. Some have treatments 4, 5, and 6, but you never see treatment 1 in the same block with treatment 4, for example. Now, there may be some unavoidable reasons why you would have to set up something this way, but I would not recommend doing it intentionally because it's usually worse than a balanced incomplete block design because it's harder to estimate the difference between means for treatments that never appear in the same block together, like the difference between 1 and 5, for example. You can still, but it's just, yeah, this way should be avoided in most cases. Another one you'll see a lot in ag science is the augmented design. In the augmented design, we have some of the treatments are called controls or checks, and they are replicated in a standard experimental design like an RCBD, like a randomized complete block. Like in this example, your orange ones, the checks, check 1 and check 2, are in an RCBD, right? Because every block has both the checks, check 1 and check 2. So checks are in a randomized complete block, but all the other treatments are either unreplicated or have fewer replicates. In this example, I put genotype 1 through genotype 40-something, only 1 each. And we can get away with this in the augmented design because the errors from the checks are used to evaluate the unreplicated treatments. So we can actually pack in quite a few genotypes without a high amount of replication, as long as we replicate the checks. So this is very common, like I said, in breeding trials where you have lots of genotypes to evaluate. I'll kind of skip over this in the interest of time. Let's see, and this is just a point that we have talked about these blocking stuff in space, but it doesn't have to be spatial layout. Like, if I know a lot of you do growth chamber studies, well, experimental runs or batches can also be considered blocks because they're closer together in time, right? Like, you don't have enough growth chambers to do tons of replicates all at the same time in different growth chambers. You might only have a couple of growth chambers, so you have to do different runs over time. And you also have to think about how you allocate your treatments to those different runs and what type of blocking design you're going to use, just the same as you would if it was done in space. So the same logic applies there. Let's see, I'll skip over, yes, oh yes, another statistician encouraged me to include this slide, so I won't skip it over. A lot of times we sort of set up our block design to account for any environmental difference in the means between the blocks. But what if not just the means differ between the blocks, but the relative difference between the treatments also varies by block? That's like a treatment by block interaction. And most of these designs I've shown you do not allow you to estimate treatment by block interaction very easily because we don't have treatments replicated within blocks. And so some statisticians, like some of my colleagues, recommend replicating at least one of the treatments within each block to estimate that interaction. It's very uncommonly done. Like, if you do that, you'll sort of be the only one, but it is it can actually give you a better idea of environmental variation if you do a little bit of replication of treatments within each block as well as among the blocks. Something to talk about or to think about in the future anyway. Yeah, how do folks feel? Do you want me to keep talking for 5 or 10 minutes and finish things up, or what?
So I'm going to make I'm going to check in with everybody. I have time to stay a little bit longer, 10 or 15 minutes over the top of the hour. Anybody else, if you want to stay, you're welcome. But if you cannot, I understand, and it's fine if you need to drop off. But I will strongly, strongly encourage and recommend everybody to stay for the common mistakes, since we can all make mistakes, and this will be good for everybody to hear.
Thanks for that. Well, yeah, like I said, this is the first time I gave this talk, so I may, in future iterations, cut out a few of the slides, but I appreciate you all being the guinea pig for this. So yeah, I'll just keep talking for a few minutes, and I won't be offended if anybody has to quit and leave the meeting. Yeah, so these are some of the common experimental design mistakes I see. I'll go through each of them real quick. Well, right, we were talking about blocks. The whole point of blocks is that units within each block are supposed to be more similar to each other than they are to things outside of that block. But a lot of times I see block design set up like this, like each block is a super long strip. And this is not great, right? Because block, kind of the word block implies a spatially compact thing, and everything within it should be spatially closer to each other than it is on average to things outside of that block. Well, that's not the case with this setup, right? Because, like, if you're in this corner, you're just as close to observations outside your block. You're much closer to these ones than you are to ones in your same block that are way over here. So that block is not going to represent homogeneous areas in the environment. So try to set up your blocks to be like as square as possible and not long and skinny. That's something I see a lot. Another one, which I think people should be aware of, is the idea of pseudo replication. That's when you treat multiple observational units within the same experimental unit as if they were independent, even though they're not. Like this study I had to help out with recently, right? We're looking at eggs laid by chickens in free range conditions versus cage conditions. Well, but we only have one barn that's free range and one barn that's caged. Yes, we have a lot of chickens in barn 1 and barn 2, but those chickens are not experimental units. They're all from the same barn, which is sort of our unit. And yes, we have a lot of observations, but those are not independent experimental units. If we treated them as independent units in our statistics, we would get incorrect inference about the difference between free range and cage, right? Because we don't know if there's just something different about this one specific barn besides the fact that it has free range that is making the chicken's eggs have different characteristics. So we call that pseudo replication. Yes, it stems from a confusion between experimental unit and observational unit. And this is related to the issue of confounding. Confounding means the inability to distinguish between different influences on our response. And in this case, we can't distinguish between the effect of free range versus cage from the effect of this specific barn, that has free range, So that might be okay. And you can still get valuable information from these unreplicated studies, but you have to consider what population you can make inference about. Like we could treat these chickens as independent units and make a very strong inference about the difference between this specific barn from this other specific barn. But, yes, you could do that, but is that really what you care about, right? Because we want to make an inference about the population in general of free range barns so that we can make general statements about, well, eggs from free range chickens are this way versus that way. So yeah, you can't do that if you don't have replication. Yes, Lena.
So like, for example, for us, like many of us do experiments with plants, like if we were to think about it with plants, like instead of a barn, let's say it's a corn plant.
Yep.
And instead of free range versus cage, it's like infected versus non-infected. How would you think about this pseudo replication situation in that case?
Yeah, well, yes. I mean, if you took a bunch of cells or aliquots or whatever from the same plant that was infected versus uninfected, those are not independent of each other. You could not treat them as replicates for the purpose of your study, because then you're not comparing infected corn plants population versus uninfected corn plants. You're comparing this specific infected plant from this other specific uninfected plant, right? Like, again, I still think it is valuable to publish data from unreplicated studies, right? Because they contribute to our knowledge overall of the topic. It could go into a meta-analysis. It's still data that's important. You just shouldn't, really, you shouldn't be making life or death decisions off of the outcome of one experiment, even if it had replication anyway, right? Because who knows if that's like reality, right? You really should be looking at the totality of the evidence on a topic before you make billion-dollar decisions. Like I wouldn't do it even if it had 10 replicates each, right? So like I would want to look at a lot of experiments before I did that. And it's just like even more extreme if you only have one replicate per experiment, right? Does that make sense? Yeah, and another point I wanted to mention is I've seen this kind of thing a couple of times, is inadequate randomization in an experimental design. Like I've seen this, people are like, okay, here's my randomized complete block, and here's block 1, across the top, then here's block 2, block 3, and block 4. Well, what's the problem here, right? They probably did not randomize the location of the treatments within each block. They might have. It could have just come out that all treatment 1 was on the left side, but probably not. And that's okay if you do it that way. If you replicate the whole thing multiple times and have like that entire set of blocks replicated multiple times, but usually it's not. And the problem here, right, is that like you're confounding spatial variation with treatment variation, right? Like what if there's a gradient of soil fertility from left to right on here? Well, that's going to be confounded with treatment. We'll never know if the mean of treatment 1 is different from treatment 4. Is that because of the actual difference between the treatments, or is it because of some environmental gradient? We won't know. So like people have given me designs like this and I'm like, well, you can kind of say something, but you'll never be able to, like, if a reviewer comes and tells you, well, it's confounded because you didn't randomize, they're right. And I can't really argue with that. So, definitely run the design by me before you go and plant out the thing instead of after, right? Because it's kind of too late at that point. Yes, so I have a couple slides here about statistics for design experiments, which I'll just run through pretty quickly. I've mentioned the idea of statistical power. We've talked about it a few times. That's the probability that you'll be able to statistically detect whatever effect you're trying to detect if it's real, right? Like if that's the probability of getting a true positive, in other words. And it depends on your sample size. It depends on your experimental design, which hopefully I've showed you. And it depends on what effect it is you're trying to estimate. How big that true effect is determines how likely we are to be able to detect it in our statistics. And we usually set up our experiment around getting a desired level of statistical power. And when it comes to power, you always have to balance between false positives and false negatives, right? There's always trade-offs. Like if you make your statistical test very sensitive and very likely to find a true effect, it's also going to be wrong sometimes and tell you there's an effect if there isn't one, and the other way around. So again, trade-offs always come up when we're designing studies. Now, a very important point to note is that any statistical model we set up to fit to a data set should represent the data generating process, including the experimental design, right? So like if treatment we think is a source of variation in our outcome, treatment should be in the model. If we think block is a source of variation that might influence our result, block should be in the model. If we think time is influencing the response in any way, time should be in the model and any interactions between them. And a very ideal type of tool for analyzing data from these type of experiments is a mixed model, which includes, that means any linear model that includes fixed effects and random effects. You may know what that is, but I'll just define them real quick. So a fixed effect is any effect whose levels are fixed from experiment to experiment. Those are usually the ones we are interested in comparing. Like if I did a study in North Carolina comparing two genotypes, and then somebody else did a study on the other side of the world comparing those two same specific genotypes, the levels of genotype are fixed from those two experiments from one to the other. So those are fixed effects. Random effect is any effect that we model as being drawn at random from a statistical distribution. Like it is effects that we are not interested necessarily in comparing specific levels of that effect, but we want to account for it. So that would be like blocks in a block design. We don't necessarily care what the mean of this block versus the mean of that block is, but we want to account for the variation due to block. And the most efficient way to do that is by using a random effect in a statistical model. So kind of like the conventional way to do it is treatments are fixed and blocks are random. Usually you can't go wrong with that, right? Because like I said, if somebody else took your experimental protocol and wanted to repeat it, they would use the same treatments that you would, right? Those levels would be fixed. But they would not, or they could not use the same blocks that you did, right? Because they would be doing their experiment in a different greenhouse or different field or whatever, right? The blocks there would be totally different. We think of them as being randomly drawn from a population of possible blocks that could have been used. Like you could have done your study in the next field over, and then you would have had different blocks. So the variation is random there. So people also ask me, well, what about like time or year? If I have a model that's repeated over multiple years, is time a fixed effect or a random effect? Well, it depends on your question, right? Like oftentimes it might be fixed if you are specifically interested in comparing the means from different years. But if you just repeated a trial over multiple years just to get an idea of the variation, well, each year is kind of a one-of-a-kind, idiosyncratic set of environmental conditions. We could never reproduce those exact conditions again in the future, even if we wanted to, right? We don't have a time turner like Hermione from Harry Potter, so we can't go back in time and do that experiment in the same exact environmental conditions of that time. Each year is one of a kind. So in that case, we would say year is a random effect, right? But I think that is the most important thing. Yes, it's important whether we decide to have year as fixed or random or whatever, but the most important thing is to make sure that all relevant influences on your response variable are included in your statistical model, whether you do an experiment or an observational study or whatever, right? Like if year is fixed, year is random, yes, it matters, but what matters more is including year at all. Like if you left it out entirely, that would be a much worse consequence. And like I said, if you ever are unsure, you should always ask me or another statistician, and hopefully we can help you. Here are some packages in R and some procedures in SAS that you can use. R has this thing called task view, which allows you to look at all the packages on a particular topic. And there's one for experimental design. These are some packages that allow you to design experiments and evaluate statistical power for different experiments, and similar functionality is in SAS. So let's end real quick, since I'm a little over time, with some kind of overall recommendations. But first, I wanted to, since I've been kind of a cheerleader for experiments this whole time, I want to just give a caveat of kind of they are not everything, right? Like this guy here, see this guy firing up a pipe in this picture is R.A. Fisher, the guy who I was like, hey, that guy's cool. He invented experimental design. Well, in his later years, he became a paid advocate for the tobacco industry to discredit any research that was at the time trying to establish a link between tobacco use and, cancer and other diseases, right? Like this tabloid tried to show that, well, yeah, smoking does not cause cancer. And he did this by, I would say, weaponizing the idea that, well, correlation does not imply causation. We can never know about if X causes Y unless we do a manipulated and randomized controlled experiment. And he sort of exploited that bias to discredit anybody trying to link tobacco use to disease, because even now we don't really have randomized trials where we force some people to smoke and some people not, and all else equal, right? Like it's sort of based on observational associations. But yes, I would say this is the reason that this is not the right view is that just because a randomized trial is the best or the gold standard way to conclusively establish causality does not mean it's the only way or the best way to obtain knowledge. We might have no choice to get knowledge in other ways, and we can still learn a lot about the world without doing experiments. And especially nowadays, a lot of new statistical methods have come up that are better at inferring things about causality from observational data if we properly control for the confounders. We can actually get a lot closer. Yes, it's not as perfect as an experiment, but it gets us a lot closer to that understanding, even from observational data. And again, another caveat I just want to throw out there, always keep in mind, I said at the beginning, even if an experiment conclusively establishes that one thing causes another within that controlled conditions of that domain of the lab or the greenhouse or wherever you did the experiment, those are artificial. It still doesn't prove that that's going to be the case in the real world. You still have to have real world observations as part of the scientific method. Experiments are important, but they're not all there is. So I'll end with my recommendations, or 10, or it might not quite be 10, but commandments for effective experimental design, and then I'll let you go. When you're designing an experiment, break it down into those treatment structure, design structure, and response structure parts. That will help you figure out how to set things up. Always be explicit about what question you're trying to answer, whether that's a conceptual question or the specific quantitative statistical question you want to answer. Write it down, because that will help you design the experiment accordingly. Again, experiments strike a balance between realism and control of variables. Think about where you want to fall on that continuum. But, don't overdo it with interactions unless you're prepared to deal with the complexity of interpreting all those interacting factors. Think outside the box of randomized complete block design. Be creative. Consider alternative designs which might be better for your situation. And if you can, it's always better to do a formal quantitative power analysis, evaluating those different designs and seeing if we can rank them on which is best in terms of statistical power. And yeah, if you know how your data will be structured and how you will analyze them before you do the experiment, a lot of the work is kind of already done before the study actually starts, and it makes it a lot easier for you. And whenever you are unsure, consult me or your friendly local statistician for help. Thanks very much. And I've also included some references for further reading if you want, and I'll like I said, put the recording and the slides up on the website, and I'll send you all the link to that as soon as I can. So yeah, thanks for your patience and thanks for listening. I'll stop the recording.
All right.